Okay, so let's start the class. In the last class what we were discussing was we were talking about the development of the male gametophyte, and whenever we use the word male gametophyte, what we understand, we are talking about what? We are talking about the pollen grain, right. So, where does the pollen grain develops? It develops inside the anther, and what is the process? We have already discussed, right. Talking about the pollen grain, we have talked about the germination of the pollen grain, division of the pollen grain into two cells, the vegetative cell and the generative cell. We have drawn the diagrams, pre-pollination and post-pollination. So, we will be continuing from that point. Below the diagram, aji 3 point dim, hey 3 point tumalke likhi lua. ठीक है sir? I think you all have drawn that diagram, no? Pre-pollination and post-pollination. 5. I think it was from A to E, right? So, below that, just continue. Point buli likhi ba. As far as the explanation is concerned, I think we have already explained the process, right? Last diagram to buza nai, bujai din. Ha, ekin likhi lua. These points are related to the explanation that we have done in the last class, right? Ekhini point to koisilu, but we have not written it down, so we are writing it down today. Bicelled condition we understand, right? In majority of the angiosperms, bicelled pollination is seen, but there are certain examples of angiosperms where it gets pollinated or it gets shed in a 3-celled condition also. If you are talking about 3-celled, what would be the cells? The tube cell and the 2 male gametes, right? Okay, write it down. 4th point, pollen tube likhiba. Is this over? So, as we have already discussed now, we have talked about the position of the tube nucleus. So, where will you find the tube nucleus? At the apex or at the tip of the pollen tube, right? What happens to that nucleus? Later it degenerates. Talking about the growth of the pollen tube, it is chemotropic and apical. What we understand by the word chemotropic and apical? Apical is it grows by the apex, right? It is only the apex that keeps on growing, right? So, that is called as apical. What is chemotropic? With the help of certain chemicals. We will be talking something called as pollen and stigma interaction. Okay, pistil stigma interaction. At that time we will be talking about certain types of chemicals that are synthesized by the style so that the pollen tube can enter inside the style and go for fertilization, right? জেনাৰেটিভ সেল ডিভাইড কৰি পেলাই দুটা মেল গ্যামেট ফৰ্ম কৰিব আৰু টিউব সেলত এটা টিউব নিউক্লিয়াছ আছিলে হেতুৱে কি কৰিব একদম এট দ্য এপেক্স ইট উইল গো এণ্ড ষ্টে দেয়াৰ এণ্ড আল্টিমেটলি ইটস গোইং টু ডিজেনাৰেট right so that is the reason why in that diagram number 5 last of ami tube nucleus to ki degenerated form ot rakhisu right oh male gametes ko prai he generative cell to divide koribo okay next point is something about the pollen tube we need to write absorptive and absorptive is absorptive okay So, this is a description of the pollen tube that we have drawn in our last class, right? Last class at point diagram number E talks about the pollen tube, and the pollen tube has three zones: the growth zone, the nuclear zone and the vacuolization zone, where we see the presence of callose plugs. What is the function of callose plugs? It separates the older cytoplasm from the newly formed cytoplasm. Where will you find the newly formed cytoplasm? Always at the apex or at the terminal position of the pollen tube. What is the reason behind? Because the pollen tube always grows in apical direction, or it has a apical growth. Jete apical growth ase apex ot grow koribo to enough cytoplasm kot thakibo, apex ote thakibo, right? So what is the function? They will separate, right? Okay. So, up till here what we have discussed is we have talked about the male reproductive part. We have talked about microsporophyll and the process or how the pollen grains are formed. We have talked about the male gametophyte and the germination of the male gametophyte. Next is we have to talk about the female reproductive structure. So, heading koriba megasporophyll. Eto hua nai? Okay, likhiba. If this is over, please write megasporophyll. So, in case of angiosperms, what is megasporophyll? The megasporophyll boli kole kuntu part of koeso? Okay, the whole part, the carpel or the pistil, right? Because we are talking about a leaf-like structure, and what is that leaf-like structure? The carpel or the pistil. So, megasporophyll is equivalent to dash in case of angiosperms. Megasporophyll is equivalent to carpel or pistil in case of angiosperms, because it is these words, that carpel or pistil, these are the words that we hear only in case of angiosperms. But if you are talking about words like microsporophyll, megasporophyll, we have used these words from pteridophytes itself, right? Is this over? So, what is a megasporophyll? The pistil or the carpel is called as megasporophyll. As far as the parts of a carpel is concerned, keta part thake? Tinta, kiki? Stigma, style and ovary, right? So, this is something that we have already discussed in morphology. This is a portion that we have already discussed during our morphology class. This is just, I am writing it down as a briefing point. It's a portion to bonot ase second. Polycarpillary or multicarpillary, no? And if you are talking about multicarpillary, remember the condition, the free condition and the fused condition. Okay. So, if you are talking about the megasporophyll, we are talking about carpel. Why we have to talk about carpel? Because we are talking about sexual reproduction. A flower has a female reproductive part which is called as gynoecium. Gynoecium, we know, individual member is called as a carpel or a pistil. A flower can have a single pistil or it can have many pistils. And if we are talking about multipistillate condition, all the pistil might be free or all the pistil might remain fused with each other. And since we have two different words, those two words are important from morphology point of view also. right? Apocarpus, syncarpus, word gita ami janibo lagibo. And talking about stigma style ovary akibo lagibo niki? Right? So, we will write certain points about it. It is a briefing, ठीक है सर, जाते नहीं कि तो बांके मोनोट रखा words किटा, क्या तमाम points लिखा, Okay, do you remember this line from morphology? Since we are talking about the reproductive part, we are talking about ovary. What we see inside the ovary? Inside the ovary, ami ki rekha pao? We see chambers of or locules. Remember the words called as unilocular, bilocular, trilocular, etc. What exactly we are talking about? We are talking about placentation, right? Because inside the ovary, what you find is the ovules. Where will you find the ovules? That study in the placenta, that study is called as placentation, right? And if you remember from the placentation point of view, which placentation is supposed to be the most advanced placentation? Basal placentation is the most advanced placentation. Basal placentation क्यों advanced? Karon, one of the reason behind that is first it's found in the advanced family. Second is if you are talking about the number of ovules, reduction in the number of ovules tells us about a what advanced character. In one ovary, ovule keta eta ovary bhitor ovule keta bahutta. So kuntu advance hobo, obviously eta jot thakibo tat advance hobo. So basal condition ba basal placentation, what is the number of ovule that you see? It's just one. That is found at the base of the ovary, right? Since we are talking about the female reproductive part or carpel, we know that in a carpel we see 3 parts, that is stigma, style and ovary. Where will you find the ovule? Obviously inside the ovary. Some points related the ovule and the ovary, we have written it down. So inside the ovary what you are going to see? Some chambers or locules, we have already discussed, right? And inside the ovary thakibo ki ovule, ovule get converted to what? The seed, and the ovary will get converted to the fruit. Now ovule has some other names also. Ovule is also called as Megasporogenesis, or it is also called as integumented Megasporogenesis. The perfect word for ovule is integumented Megasporogenesis, because we are talking about angiosperms, and also in case of gymnosperms we have seen that the ovule has or remains surrounded by certain types of covering, and that covering is called as integuments, right? As far as Megasporogenesis is concerned, Megasporogenesis to ami beleg plants to paisu. Pteridophytes also we have seen Megasporogenesis, right? In bryophytes also we have seen Megasporogenesis, right? Kintu yate ki hobo je, sorry, in gymnosperms and pteridophytes we have seen the word Megasporogenesis, but in case of angiosperm Megasporogenesis is present, but the proper word is integumented Megasporogenesis. Karon tat integuments ase, pteridophytes jintu Megasporogenesis asile tat integuments nai. Okay? So these are certain informations, and the next thing that we have to draw is the structure of the ovule. So heading korba structure of ovule. Likhilba. As far as anatropous ovule is concerned, we will be discussing. While we were discussing gymnosperms, no? I think we have talked about certain words called as autotropous and anatropous, if you remember. Gymnosperms are, what is the type of ovule that you see? Autotropous, that is also called a straight ovule. But in case of angiosperms, we have a word called as anatropous. What is anatropous? Inverted ovule. So we will be talking about it. Is this over now? So structure of ovule kora, just to have a rough idea how does the ovule look like. Raphe, RAPHE. Tinta antipodal cells, tinta egg apparatus. So, if you are talking about a ovule, generally we draw the diagram of anatropous ovule because that is the most common condition that we see in case of angiosperms, right. So, if you are talking about anatropous ovule, the position of the micropyle will be facing towards the ground. So, it is here. The side opposite to the micropyle is called as selagia, right. Next, talking about the parts, I think we have understood integuments, the covering. As far as integuments are concerned, we will be writing certain points. We have certain words called as unitegmic, bitegmic, tritegmic, etc., right. The most common point that you need to remember is the comparisons between gymnosperms and angiosperms. In case of gymnosperm, we had a word called as unitegmic. In case of in case of gymnosperm, we had a word called as unitegmic. In case of angiosperm, we have a word called as bitegmic. I think we understood presence of one integument, presence of two. Next, as far as this dotted area is concerned, it is called as the nucellus. What is nucellus? It is nothing but the nutritive tissues that gives the food to the growing embryo sac, right, and it is the ovule that develops from the nucellus itself. Ultimately, hey, embryo sac or ovule portion to corpora develop koribo. One cell of the nucellus towards the micropylar end will enlarge and ultimately it will form the ovule. We have already studied as to how the pollen grains are developed. No, we have talked something called as the archisporial layer, hypodermal layer, etc. So, similarly, we are going to talk about how does the ovule develop, from where does the ovule develop, etc., right. So, we have the ovary. Inside the ovary, we have that nutritive tissue that is called as nucellus. Nutritive tissues mane ki? It's collection of cells. One cell amongst the nucellus that lies with the micropylar end will become big and ultimately it will convert itself into a ovule. So, who forms the ovule? It is the nucellar cells, or it is the nucellus from where the ovule develops, right. Okay. Next is, as far as this portion is concerned, I think you have heard something called as a 7-celled and a 8-nucleate structure, right. So, this is something that we will write it later, 7-celled 8-nucleate structure, central cell, endosperm, we understand, right. Talking about this structure, this is also called as what? Embryo sac. Next, we have certain words called as funicle, hilum and raphe. What is funicle? The stalk of the ovule is called as a funicle. If you are talking about an ovule, I think you have seen certain seeds, no? Seeds or tolat ki thake, a stalk thake. So, the best example is, or most obviously seen in case of a lychee. Lychee seed to tolat horu ke thake, no? That stalk is called as the funicle. As far as hilum and raphe is concerned, what is hilum? That stalk and the ovule, juntu point at lagi thake, that is called as what? Hilum. The point of attachment of the funicle and the ovule. Since we are drawing the diagram like this, it might be a little bit confusing, but if you are talking about hilum, if this is the ovule, if this is the stalk, a portion to ki boli kom? Hilum. The point of attachment of the funicle with the ovule is called as hilum. Etal ki hoy? Funicle. Etu ki hoy? Ovule. A point to ami ki boli kom? Hilum. Right. Next, we have raphe. What is raphe? Sometimes it is seen that the funicle ki hoy, one portion of the funicle covers the ovule at a particular area. Okay, the funicle ta yat lagi thakibo lagisile, kintu okkoman grow kori overlie or surrounding the ovule. That area is called as the raphe. If you are talking about a seed, what we see is in a seed, eta portion of we see a ridge like a structure. What is ridge? RIDGE, ridge and furrow. Hunisane? Jika sini poa jika, ami juntu khau. What do you say? I don't know. It's ridge gourd. Yeah, it's a vegetable that we eat, right? It's long. What do you say? I don't know in Hindi. Ki koi? Okay, ridges and furrows means what? Ridges are two portions that comes out, and if suppose we have how to explain you? Suppose this is a ridge, this is a ridge. The underlying portion is called as furrow. The extended or the thing that is on the top is called as a ridge. Between two ridge, what you see is a furrow. Figure se niki jikar. Dekhai diya bhai. Jika boli marile kene ke hobo ridge gourd mara. RIDGE ridge, GOURD. I don't know the Hindi like meaning for that. Kukurbita? Oh yeah, family hoy. Kintu. Okay, let's not waste time. नहीं पता, देखा ही नहीं. हाँ वही होता है. उसमें क्या होता है कि you have a furrow type, low lying area and elevated area. That elevated area is called as ridge. ऊपर जो है that is ridge और नीचे में जो है ना it's like yeah that is the furrow, right? So if you see a seed, in the seed one area of a seed has a ridge. थोड़ा सा ऐसा if you see a seed, one area of a seed has a little bit elevated portion. It looks, this is the seed, बाकी जगह देखो नहीं, ए जगह के लिए অকমান এটা marking আছে, right? This is what is called as raphe. What exactly a raphe is? The extension of the funicle over the body of the ovule is called as raphe. What is hilum? The point of attachment is called as hilum. What is funicle? The stalk of the ovule is called as funicle. We have words called as micropile and cellagia. What is micropile? It is that area through which the pollen tube will enter. The first point. Second point, it is that area that is not covered by the integuments. If I are talking about the ovule is surrounded by the integuments on all sides except the area that is called as micropile, right? Funicle. You have the ovary wall. Where will you find the funicle inside the ovary? This is a description that we have already done while we were discussing placentation. Inside the ovary, we know the ovules are present. Where will you find the ovules? What is the location of the ovule in the ovary? The ovules are found wherever the placenta is present. Remember certain words called as marginal placentation. Why is it called as marginal? Because the ovules are found in the margin of the ovary. Why is it found in the margin? Because the placenta is in the margin. যদি সেন্টারত থাকিলে হয় ওভিউলস বিলাক কোত থাকিলে হয় সেন্টারত থাকিলে প্লেসেন্টা কি হয় নাথিং বাট দ্যাট কালেকশন অফ প্যারেনকাইমাটাস টিস্যু দ্যাট ইজ নিউট্রিটিভ সো ওভারি আছে ওভারিত প্লেসেন্টা আছে প্লেসেন্টাটো যতে থাকিব তাতে ওভিউল থাকিব রাইট যদি মার্জিনত থাকে তেতিয়াহলে তাত কি থাকিব মার্জিনাল ওভিউলস হ'ব যদি বেস পোরশনত থাকে তেতিয়া বেসাল ওভিউল হ'ব এটসেট্রা এটসেট্রা রাইট অ্যাজ ফার এজ ওভিউল ইজ কনসার্ন ইট হ্যাজ আ স্টক টু কোট লাগি থাকিব প্লেসেন্টাত লাগি থাকিব প্লেসেন্টা টু কোট লাগি থাকিব ওভারি ওয়ালত লাগি থাকিব right? So if the structure is over, we'll write certain points. Aapko points fully likha. Can I rub this? Okay, we are writing the points itself, no? So 1, 2, 3 we have already written. So number 4 we'll write now. Okay. Point number 4 likha. What is the integument? The covering of the ovule is called as integument. What does the integument convert into after fertilization? Seed coat. Right? I mean you have heard the words called as testa and tegmen, yes. Right? Testa kune form kore outer, tegmen kune form kore inner, right? We have certain words associated. Seed coat, কাৰণ seed coater দুটা layer থাকে ন এটা হল তোমার টেস্টা, এটা হল টেগমেন। দ্য বেষ্ট সিড টু সি দিস টু ষ্ট্ৰাকচাৰ ইজ দ্যাট গ্ৰাম সিড। কাবুলি বুট যোনটো আছে ন, এনেও বুটত দেখা পাবা, কিন্তু সৰু সৰু বিটবিলাক বেছি সৰু হয়, চ' ইটছ ভেৰি হাৰ্ড টু ডিফাৰেঞ্চিয়েট ন। কিন্তু ডাঙৰ যদি গ্ৰাম হয় তেতিয়াহ'লে তাত আমি দুখন কাভাৰিং দেখা পাম, ৰাইট? Okay, so from integuments point of view, unitegmic condition and bitegmic condition, something that's common in case of gymnosperms, something that's common in case of angiosperms, but we have exceptions. Obviously we have certain exceptions, right? The most common condition in case of angiosperms is bitegmic. We have seen an example of a tritegmic ovule also. Tritegmic is found in Asphodilus. Asphodilus is a plant that belongs to monocot, and monocot obviously belongs to angiosperms, right? So we have exceptions. And next we have ategmic, where the integuments are absent. One example is centellum, right? From these points, 8 point important. Ategmic iman question aha nai. Okay. The next point that we have to talk about is, a question is asked about nucellus. We have two types of nucellus. First, what is nucellus? The nutritive tissues, right, from where the ovule develops, that is called as nucellus, right? What happens to the nucellar tissues? As the ovule keeps on growing, the nucellar tissues keeps on degenerating, because they will be supplying their nutrients to the growing embryo sac, right? Or in certain cases what happens, apart from even after fertilization, certain amount of nucellar tissues remains in the seed. Now in normal case of ki hobo, whenever the seed gets developed, the nucellar tissues almost degenerate. So if you see a seed, you don't see the nucellus, right, because they will get generated. But there are certain examples where certain amount of nucellar cells always persist even after fertilization. Seed to usorte kisuman nucellar tissues ami dekha pam. At that time we will be using a word called as periplasmic seed. This is a word that we have already discussed in morphology. We are going to write few points about it now also, right? As far as the nucellus is concerned, we understand what is nucellus, what is the function, we understand. But here we have to discuss, the point is, have you heard the word called as cressu-nucellate and tenu-nucellate condition? Question to heteroporte ahe. Okay, I'll write it down. So what is nucellus, what is the function of the nucellus, we will not write, because we know about it, right? Nucellar cells are generally they are actually तो वो क्या होना चाहिए, diploid, right, because it's a part of the female reproductive part also, right? But we have triploid condition also, nucellar tissues, right? But mainly we know that it's a part of that sporophyte, so it is obviously diploid. tenunucellate aru crassinucellate, right. So, inside the ovary what you see is you see the nucellar tissues. If the nucellar tissues are poorly or less developed, that condition is called as tenunucellate. If it is more or well developed, that is called as crassinucellate. Question kiya hai nucellus kom thakile ki boli koy, besi thakile ki boli koy. So, you have to know the meaning of tenunucellate and crassinucellate, right. So, do not get confused between these words. So, less and more, hetu bostu poribo lagibo akon. We will continue nucellus. So, pay attention to the word persistent nucellus, the examples and the name, perispermic, persistent nucellus and the names, right. And what we have seen, one of the cells of the nucellus towards the micropilot end will become big and ultimately it will form what? The ovule. What is a megaspore? A megaspore is nothing but the ovule. What is the ploidy of a megaspore? N. Megaspore mother cell is twice N. Megaspore ki hobo? N. So, megaspore is nothing but the ovule. So, ovule bhitor thaka gute kita cells ploidy ki hobo? Understood? So, inside the ovule what you see? Certain cells, that 7 celled and 8 nucleate condition, each and every cell would be N because the entire structure itself is N, right. And we know this, no? Megasporogenesis, out of the 4 megaspores that is formed, 3 will degenerate and 1 will remain functional. Mitosis or meiosis will mitotic is nothing but mitosis. Mitosis division hole mitotic cell, meiosis division meiotic cell, right? Eketai bostu. And as far as the difference between mitosis and meiosis is concerned, mother cell producing daughter cell, all the 3 will have the same number of chromosomes, mitosis. Mother cell forming daughter cells and the daughter cells will have half the number of chromosomes as compared to the mother cell, that is meiosis. Chromosome number jodi ekei thake, tete hole mitosis, jodi half hoi jay, reduce hoi jay, that is meiosis, right. Ekhini howa nai na likhi loa. What is micropilot? Remember this diagram we have drawn, right? Etu ki hoy embryo sac, egita ki hoy integuments, right? এই জায়গাখিনিত ইন্টিগুমেন্টস নাই আছে নেকি? আমি কি বলি কম micropile, right? What is micropile? The area where the integuments are absent, first point. It is that area through which the pollen tube will penetrate. তারমানে সোয়া ইয়ার ভিতৰতে তো তোমাৰ কি আছে? egg to নাই জানো, right? So we have this, no, stigma style ovary, তাৰ ভিতৰত কি আছে? ovule. Ovule etu side by etu side. Suppose we are talking about the egg here. Yate egg ase, right? Pollen to yat asile, pollen tube develop koribo. A pollen tube to A to structure bhitor khumabo lagibo na lage, so that it can male gametes jun kita asile, male gametes kita egg logot fuse hobo lagibo. So fuse hobo lagile ei bostu to yat khumabo lagibo. So kun pin di khumabo, jedi etu micropile hoy etu sidha eneke khumabo, right? যোনটো এৰিয়াত ইন্টিগুমেন্টছখিনি নাথাকে তাক আমি মাইক্ৰোপাইল বুলি কওঁ। দ্যাট ইজ দ্য এৰিয়া য'ৰ থ্ৰুৰে পলেন টিউবটো সোমাই পেলাই মেল গ্যামেট ফিমেল গ্যামেটৰ লগত ফিউজ কৰিব, ৰাইট? If this is over, the next topic would be types of ovules. Heading কৰিবা types of ovules. এইখিনি হ'ল নেকি? নাই হোৱা, লিখি লোৱা। সিডৰ পৰাইতো ফ্ৰুটটো হৈছে, বিটটো আল্টিমেটলি আমি যোনটো পৰ্চন খাওঁ, অ'কে, আমি যোনটো পৰ্চন খাওঁ বিট এজ আ ভেজিটেবল, সেইটো আমি চিড ফ্ৰুট সেইবিলাক খাওঁ নেকি? ফ্ৰুটছ খাওঁ, কিন্তু ডাজ দ্যাট মিন দ্যাট প্লান্ট উইল নট হেভ ফ্লাৱাৰ্ছ? ফ্লাৱাৰ্ছ থাকিব নাথাকে? আমি নাখালোঁ বুলিয়ে নাথাকিব নেকি? Are you understanding? Okay, best example, another carrot, radish, এইবিলাকৰ ফুল আছে নাই? Oh, the entire plant will be root itself. It will have no leaves, no flowers, no stems, nothing. অকল root টোৱে নেকি? থাকিব নাথাকে গছ এডাল যিহেতু তাৰ ৰুটটো থাকিব ৰুট থাকিলে ষ্টেমছো থাকিব ষ্টেম থাকিলে লিভছো থাকিব যদি এনজিওস্পাৰ্ম হয় তাত ফ্লাৱাৰ্সো থাকিব যদি ফ্লাৱাৰ্স আছে সিডছো থাকিব সো ক্যাৰেট ৰেডিছ এটসেট্ৰা সিডছ আছে নাই obviously আছে কিন্তু আমি নাখাও it's not like this that every fruit we eat as a edible part it is not no sometimes the root is edible sometimes certain things stem is edible for example if you're talking about sugarcane the stem is edible if you're talking about mango the ovary is edible right fruit to edible hoy kintu if you're talking about carrot radish etc the root is edible we eat a specific part that does not mean rest of the parts are missing in that plant hoy na thik teneke beetor ami kuntu part khau root to khau kintu does that mean it will have no flowers no seeds etc korpra he plant dal develop korile seed or pry to develop korise so that seed would be an example of perispermics okay not the part that is edible okay পালেং শাক খাই পাইছা নাই স্পিনাচ গুটি আছে নে নাই গুটি আছে seeds present or absent present no sure no have you seen the seed no because we don't eat the seed কিন্তু পালেংটো গজাবলে হলে if you want to germinate কি আনা দোকানৰ পৰা কিনি পেলে seeds এটো আনো no it's not the plant that you bring and you put it down in the soil no it's a seed so obviously we have the plant has seeds but the edible part is not the fruit or the seed the edible part is the leaves in case of spinach the edible part is the root in case of carrot and radish right Okay, so we are going to talk something called as the types of ovules. Types of ovules on the basis of what? On the basis of the degree of curvature. Degree of curvature means rotation, right? Movement. Clear? What is important? Most primitive. What is important? Very common in gymnosperms and the word straight ovule. Polygonum is not a gymnosperm. Common in case of gymnosperms. Okay. Is this over? Autotropous? Understood? So we are talking about types of ovule on the basis of degree of curvature or on the basis of move. Yes, please say. Okay, okay. Ekhini? Agor kini likhisilan nai? Okay, ekhini likhilwa. Diagram to? Okay, likhilwa. So autotropous ovular key point monot rakhibo লাগিব, it's a straight ovule, right? So since we are talking about types of ovule on the basis of curvature or on the basis of movement, on the basis of what is the angle that they may make, we have to remember the angles also. So question has been asked about the angles. So question to keneke ahise, a ovule with 180-degree curvature is a dash type of a ovule. 180-degree hole ki hobo? Anatropous. Straight hole to straight hole. Next we will be talking about 90-degree curvature, right? Similarly teneke bahi jabo, and finally we will have 360-degree curvature. Straight thakibo 90-degree hobo, 180-degree hobo, 360-degree hobo. 360-degree hole ki hobo? Upside down hoi jabo na jay. It's totally upside down. Okay. Straight line ekane angle form ei kora nai. Ovule to keneke move korise hetuthe angle to form koribo, right? Clear? Huh? Water plant, huh? Heterophily is a word that we have to remember in case of ranunculus. If you, I think we have discussed ne nai koa heterophily concept or kotha koisu niki? Koisilo no? So heterophily, a plant having two different types of leaves is called as heterophily. Phily is leaves, right? So whenever you read about ranunculus, first water plant, hydrophyte, second shows heterophily. What is heterophily? An example of plasticity. What is plasticity? Changes because of the environmental conditions is called as plasticity. This plant will change the morphology of the leaves depending on its environmental condition. The part that is above the water will have a different morphological structure, the leaf, and the leaf that is inside the water will have a different morphology. That is what is called as plasticity. That is also called as heterophily. So 90-degree curvature, more or less than 90-degree, 180-degree, these are things that we have discussed. You don't have to remember any curvature this time, but the word horseshoe. Right. If you see up till this one, no, we have already discussed 4 types of ovule. First, autotropous, anatropous, hemitropous and campylotropous. In all these 4 conditions, if you talk about the embryo sac is not curved, right. It might be slightly bent or it might be 90 degree, but it is not curved, right. But in this case, it is totally curved, no, it looks like a horseshoe. So, this is the particular word that you need to remember. So, these are the different types of ovules on the basis of curvature: straight ovule, 180 degree ovule, 90 degree ovule, 360 degree ovule and a horseshoe shaped ovule, right. So, these are the things that you have to do. AC thakile to family hoy no. AC থাকিলে suffix to যদি AC থাকে, else থাকিলে কি হবো? ALES, then it's order. So AC থাকিলে it's a family, right? So here what you need to remember, 360° and the word coiled. Who coils around the ovule? It is the funicle. Edal hai funicle. Normally কি asle straight, it covers here, right? So originally or at the very beginning, this ovule is key. Or the question to ahe, in case of Sarcinotropous ovule, the ovule is straight ne anatropous. Prothomote ki asle straight. When will you call a ovule as a straight ovule? Whenever the micropyle faces upwards. In this case also the micropyle faces upwards. It's different that the funicle covers the ovule. Kintu micropyle position code is upwards looking, right? So in case of Sarcinotropous, the ovule is which type? Orthotropous or straight type. Clear? So these are the different types of ovules. What is important is the examples and the angles. Especially examples so iman important nohoy, angles bilakor aru naam gita important. Yeah. আহিছে 90° আহিছে নেকি নে 360 কিবা এটা আহিছে কিন্তু it is not the examples that are very important. What is important? The name and the respective angles, right? If this is over, points likh him. Eto hol diagram. Okay. In the meantime, we'll write certain points. Okay, if you talk about fruit, the part that we eat varies from plant to plant, right? If you are talking about the edible part being an ovary, example ki hobo mango, ovary to ami khao. If you talk about the edible part as a thalamus, apple, strawberry, etc. If you are talking about the edible part to be the third integument, aril, right? Aru khodai aril buli kole one and only example that you remember is lychee. ठीक है बेलेग एकु नाले क्वेश्चन आहे द एडिबल पार्ट ऑफ लीची इज कॉल्ड एज एरिल. What is aril? The third integument. Sometimes we see the presence of third integument in case of certain plants. Since we are talking about ovule, we are talking about the seed, right? So, we have structures called as aril, we have another structure that is called as Karangal. What is Karangal? The Karangal is nothing but a proliferation of the outer integuments. Proliferation means outgrowth, right? You have the outer integument. For example, if you are talking about an ovule like this is the inner and this is the outer, right? What happens to the integuments after fertilization? These two integuments ultimately will become the seed coat, right? Sometimes what happens in the seed, no, eta portion eneke ulai thake, right? This is called as Karangal. After fertilization, the ovule converts itself into a seed. In the seed, just in the micropilot area, right, you see certain outgrowths coming out from the seed, and that outgrowth is called as Karangal. Karangal ki hoy? Outgrowth of the outer integument, right? Normally after this, no, when this after fertilization, you should have seen a structure like this with the seed coat. No proliferation, no outgrowth, nothing. But certain seeds, you will see the presence of outer integument, the traces of the presence of outer integument. That is what is called as proliferation or outgrowth. A structure to kami ki boli ko Karangal boli ko. Karangal thakile seed to kami ki boli ko Karankulate seed. Give an example of a Karankulate seed is a question that is asked, right? Iman details na lage, right? What is Karangal? Which part forms a Karangal? Not important. The only point that we will remember is example of a Karankulate seed. Perispermic seed is something that we have already learned. Examples we know, coffee is the best example, meat and all. Now we are talking about a Karankulate seed. Karankulate seed, one and only example that you remember is castor, right? Next, since in the Karangal, what you have is sugar, right? So obviously insects bilak ahibo, and ultimately it will help them in pollinating. Pollination by help of ants is called as Myrmecophore. We will be talking about pollination little later. All of these are types of pollination to ami porhime, right? So these were certain informations about the megasporophyle, and to be precise about the megaspore, right? Ovular, different informations we have learned. The next part of the lesson we need to talk about the process. What happens to the ovule, who forms the ovule, out of the 4 megaspores, what happens to the 3, and which remains functional would ultimately convert itself into the embryo sac. So, the next portion of discussion is something called as Megasporogenesis. Microsporogenesis we have already done. The formation of microspores is called as microsporogenesis. Now, we will be discussing Megasporogenesis. What is Megasporogenesis? The development of megaspore is called as Megasporogenesis. Who forms the megaspore? The megaspore mother cells forms the megaspore. Who forms or who behaves as the megaspore mother cell? Nucellar tissues that lies towards the micropilot end would behave as a megaspore mother cell. So, heading koriba Megasporogenesis and the female gametophyte. Is this over? Ekini hol? Ekini nai hua. Oh, M-Y-R-M-E-C-O, Myrmecophore, C-H-O-R-Y. So, we will be talking about who forms the megaspores and how does the megaspore develop, right? Megaspore develop, megaspore could also be called as female gametophyte because inside the megaspore we have what? The egg, right? So, we have the ovary, inside the ovary we have the ovule, inside the ovule we have the egg. Eketai kotha? next point বুলি নহয় heading এখিনিতে তোমাৰ হেৰি লগত আছিল পইণ্টছ আছিল ওভিউলৰ এটো প্ৰসেসটো heading So microsporogenesis and the development of the male gametophyte আমি যেনেকে diagrammatically study কৰিছিলো ঠিক তেনেকে megasporogenesis also will be studying with the help of certain diagrams, right? So if this is over, we'll draw the diagrams. How good? So diagram kita আঁকি দিও একেটাই নাম তাতো আৰ্কিস্পোৰিয়েল সেল ইয়াতো আৰ্কিস্পোৰিয়েল তাতো স্পোৰোজেনাছ সেল ইয়াতো স্পোৰোজেনাছ সেল hypodermal region of the nucellus তামানে আৰ্কিস্পোৰিয়েল সেলটো কোড ডেভেলপ কৰিব ইন দা hypodermal region of the nucellus আমি যেনেকে মাইক্ৰোস্পোৰোজেনেসিস অফ পঢ়িছিলো ন hypodermal region right epidermis asile epidermis er taller layer to ki boli kom hypodermis similarly in this case also we have the epidermis below the epidermis will have the hypodermal layer in the hypodermal layer one cell will enlarge itself and ultimately it will behave as what archisporial cell right তাতো এইটোৱে হৈছিলে মাইক্ৰস্পোৰোজেনেছিছতো স্পোৰোজেনাছ চেলটো মিঅ'চিছ হৈ পেলাই মাইক্ৰস্পোৰ ফৰ্ম কৰিছিলে কাৰণ স্পোৰোজেনাছ চেলৰ যিমানবোৰ চেলছ আছিলে দে উড বিহেভ এজ what microspore mother cell in case of microsporogenesis and here the collection of sporogenous cell would behave as megaspore mother cell in case of megasporogenesis. So each MMC will divide. Which process of division? Obviously meiosis, and ultimately what they will form? A collection of 4 haploid megaspores, right? So out of this 4 haploid megaspores, we know that 1 megaspore will remain functional and baki 3 ta degenerate koribo. I think the process is understood by the diagram itself, right. So, ultimately from one cell, that is archisporial cell is nothing but the cell of the nucellus, will ultimately form this embryo sac. Etu ki boli koy polygonum type, remember 7 cell 8 nucleate, he type to ki boli koy, that is called as monosporic polygonum type of ovule or embryo sac. Embryo sac we will be talking about later in this chapter itself, right. In this diagram what we have seen is out of the 4 megaspores that were formed, 3 of them got degenerated and only 1 became functional who formed the embryo sac. But this is not the condition in all the types of plants. Sometimes in certain plants, out of the 4 megaspores, 2 will participate in the formation of the embryo sac. Sometimes it might be like this, that all the 4 will participate in the formation of the embryo sac. But the commonly seen feature is out of the 4, it is only 1 that is becoming functional, and so we have that word called as monosporic. If we use a word that is called as bisporic, at that time we will understand that out of the 4 megaspores, 2 of them remain functional. Tetrasporic embryo sac. So, at that time we will know that all the 4 megaspores remain functional. So, depending on plants to plants, as far as the number of megaspores remaining functional is concerned, it varies. But what is the most common type that is seen in case of angiosperms is, out of the 4 types, it is only 1 functional megaspore that ultimately forms the embryo sac, right. A2 is a hobo, not necessary, pelego hobo pare. Microsporogenesis, the point to be remembered is out of all, all the 4 microspores that are formed remains functional. All the 4. Hetu sentence e monot rakhiba. Megasporogenesis 1 out of 4 and microsporogenesis all the 4. Right? Clear? Okay. Online students kiba doubts asel niki? Okay, few points. Huh? A gutei to eta cell. The large, we will be studying about this. A2 details hami porhim. The largest cell in the embryo sac. Which one is the largest cell? Yeah, so both cell is A2, 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 A2. Gutei cell hoy. Largest cell kuntu? The central cell. Central cell or nucleus keita? Duta. Bujhi paisa? Duta nucleus eta cell. Duta eta cell or duta nucleus ase. Right? Just give me 5 more minutes so that I can finish this part. Okay, points to taman likhasun. Second is a diagram. Embryo sac. Confusion kihar word ase? Female gametophyte. Gametophyte, gamete bearing plant is called as a gametophyte. Which gamete are we talking about? Female gamete. The female gamete is also called as ki egg. Egg to kot ase? Embryo sac or bhitorot ase. So embryo sac to kami ki boli kom? Gametophyte. Egg to ki boli kom? Gamete. Right? Okay, a diagram to important karon yar pora question ahe. Microsporogenesis wallখন ফর্ম কৰিছিলে Megasporogenesis wall ফর্ম কৰিব কিন্তু single layered তাতে 2 to 6 layered আছিলে so since it's a single layered so we don't give stress on that right A diagram to kya karne aakise because the question has been asked if you are talking about the development of the functional megaspore first question where will you find the functional megaspore obviously inside the ovule. Second, if you are talking about ovule, you have two particular area. One is the micropyle and the other is the selagia, right? The question is based on this fact. Functional megaspore would be present on which site? Micropyle thakibo ne selagial area thakibo. That is the reason why I am doing this diagram. Right? We know that we have already talked about archisporial cell. Who forms the archisporial cell? Any cell of the nucellus towards the micropyle will enlarge itself and ultimately it will form a archisporial cell. This archisporial cell will divide into two cells. We have discussed parietal cell and a sporogenous cell. So, parietal cell kot thakibo micropyle, sporogenous cell kot thakibo towards the selagia. Clear? Again, sporogenous cell will divide and form 4 cells. Yes or no? 4 megaspore. So, ultimately next diagram to kenekwa hobo. Something like this? Yes or no? Clear? So, this was A, this was B, this is C. Now, we know out of the 4 only one will remain functional. The question that is asked is functional megaspore to kot thakibo selagial side or ne parietal selagial side ne micropyle side ne selagial side. We have seen a row of 4 microspores. এইটো ডায়াগ্ৰামত কি দেখিছে এটা প্যাৰাইটেল 4টা মাইক্ৰোস্পোৰ আছে মেগাস্পোৰ আছে নাই সো এই 4টাৰ ভিতৰত এটাহে ফাংশনাল হ'ব কোনটো ফাংশনাল হ'ব এই 4টাৰ ভিতৰত that is the answer to this diagram. It is the one that remains to the selagial side will remain functional বাকি গুটেই তিনটা degenerate কৰিব right. Clear? So, এইটো পৰ question আহিছে functional megaspore to কোনটো সাইডে থাকিব this is the selagia this is the micropyle হা এইবিলাক তো পিছত সব ডিজেনাৰেট কৰি যাব না গুটি খোপা ডিজেনাৰেট কৰিব থাকিব কি অকল এইটো so everything 1 2 3 and the parietal cell ultimately everything will degenerate. So, this was the second one and finally we write the last point of today's class the third point. So, just a question that has been asked is peramylar bodies is found in, it is found in lilium. What is peramylar? You do not have to remember, right. Now, we understand what is a peramylar body. It is a storage area that contains what? Carbohydrate, right. Coat in between cell wall and plasma membrane of which cell? Megaspore mother cell in case of whom? In case of lilium or lily, right. So, only this one and this one, that is all. So, today I think this is all for today. In the next class, heading to lichidoidium, development of the female gametophyte. We have to talk about how does the embryo sac develops into a 7 celled and 8 nucleate structure, right. This is something that we are going to discuss in the next class. Thank you.